Hi, welcome everybody. Welcome to the 2024 L. Jackson Newell Fellowship presentation. I'm Monisha Pashupathy. I'm the Dean of the Honors College. It's fabulous to see all of our students and faculty and guests and others here. I'm really thrilled that you could be here with us. I'm just going to say a couple words about what this series is about, and then I will hand it over. And then I will hand it over to Professor Christopher Miller, who is an honors faculty member who chaired this year's selection committee. So this lecture series was initiated by honors faculty. And I want to say a couple words about why honors faculty are particularly interested in this. Is that better sound-wise? I feel it's what? It's going in and out? Okay. Give me I'm looking back at the tech folks. It's just, I just have to focus on, okay. Human error, it's a thing. So honors faculty train in their disciplines, just like faculty at the rest of the university. They specialize. But when they choose to join honors, they're committing their capacities towards educating students in a foundational liberal arts and sciences curriculum. So their desire to launch this lecture series is grounded in their appreciation for the liberal arts and sciences, for the foundational and enduring value of those liberal arts and sciences, and for the ever-increasing importance of interdisciplinary understandings that is fostered by that broad foundation. So the idea behind this lecture came from honors faculty, and it was to honor, recognize, and celebrate faculty from all over the university whose work demonstrates that enduring value and continued relevance of work in the liberal arts and sciences. They also wanted and hoped to spark a set of common conversations across the university around that work and the faculty member being celebrated. So that was the initial goal. And then we had this problem of what to call this series. And so as we designed it, this question of naming arose, and faculty proposed and quickly coalesced around the idea of naming this series for L. Jackson Newell. Jack Newell, as many of us know him. So Jack Newell is an expert in the philosophy and history of higher education. He was faculty here at Utah in the College of Education for many years, but he also served as what was then called the Dean of Liberal Arts at the University of Utah. So in that role, he built an award-winning program that now you might know as a general education program. And that program that he built actually remains at the core of the honors curriculum. Sorry. Jack spearheaded access to liberal arts foundations in the community among non-college bound individuals and vulnerable youth in programs that are still running. He also served as the president of Deep Springs College, which is a very innovative experimental two-year college located in the Sierra Nevada desert, in which students both run a working ranch and also complete a liberal arts foundation curriculum. And they engage in self-governance. They are part of the way the whole college runs. So Jack was emblematic of this value in liberal arts and sciences. His whole career was around supporting liberal arts and sciences. And so it made total sense to name this lecture series after Jack Newell. So that's the context. But every year we choose a Newell Fellow, and the real point of this evening is to celebrate the work of that fellow. And so I'm going to turn this over now to Christopher Miller, who can tell you a little bit about this year's Newell Fellow, and then we'll get on with the real best part of the show today. Thank you.
Let's see if I can do this. Can you guys hear me all right? All right. So as Dean Pasupathy mentioned, my name is Christopher Patrick Miller. I teach in the Honors College. Hopefully I'll get to meet some of you guys. I served as the committee chair for this year, and I'm excited to introduce Professor Michael Werner, or as you might know him, the worm wrangler of the U. I think everybody watched that video. Who is our L. Jackson Newell Fellow for this year. Professor Werner's work emerges out of a biology lab here on campus, the Werner Lab, that has served as a real incubator for innovative scientific research. The Werner Lab currently has two, let's say, broad concurrent projects, one of which we'll hear about today. And the other project is on the epigenetics of phenotypic plasticity, which from what I gather, aims to figure out, and this is a direct quote, the identity of epigenetic information carriers that contribute to alternative phenotypes and determine how these modifications communicate environmental information into transcriptional and ultimately physiological and morphological phenotypes. I'm not going to pretend to understand exactly what that entails, but it sounds very significant. In the spirit of the Honors College, Michael's lab regularly supports and mentors undergraduates in the research process, regularly incorporating students in his lab through the Science Research Initiative. Students have served as co-authors on his papers, including his recent publication with the Proceedings of the Royal Society of Biological Sciences on newly identified nematodes in the Great Salt Lake. Michael has been a frequent visitor to a course I teach on land art in the Great Basin region. And whenever he visits, he makes a point not only to communicate the biological and geologic history of the Great Salt Lake, but also the urgent issues we face as collective stewards of this unique terminal lake and ecosystem. I can tell how impactful his talk is with students because they continue to talk about it as we travel around the Great Salt Lake, debating, for example, why Nancy Holt would take all the effort to build enormous concrete tunnels and drop them in a remote area near an old railroad. Michael helped me see how important it is that we understand the layered histories of this place we live. And to understand it, you often have to go there, be there, experience how this arid expanse shifts our sense of ourselves. When we discussed the many great proposals we had for this lecture, we settle on Professor Werner's work not only because of its scientific innovation, but also because it spoke to a larger cultural, ecological, and political conversation about the future of the Great Salt Lake. This conversation feels urgent and necessary, and we're excited to hear what Professor Werner has to add. Professor Werner.
Okay, can everyone hear me? All right. Thank you very much for that introduction. It really is an honor to be this year's Jackson Newell lecture speaker. So I'm going to tell this story today from the beginning as it really happened. And so often in science, the road to discovery is circuitous and filled with starts and stops and multiple dead ends. But when it becomes time to publish our discoveries, we reimagine some narrative of a brilliant master plan, and all the experiments that unfolded thereafter went exactly according to that plan. And it's rarely the case, but I think in this particular story, the reality of the winding path is particularly interesting. And I also think that there are potentially some lessons in here about going into nature and paying attention to it, and challenging or questioning orthodoxy and status quo, and really just being curious. And I think those values are embodied by Jack Newell, and so I thought it was particularly appropriate to tell the story as it was. So this story begins in 2020. I'm finishing my postdoc at the Max Planck Institute in Germany, and my wife and I both got offers in the School of Biological Sciences here at the University of Utah. And so I was really excited to come back to the States and open my lab. And the main question that my lab seeks to answer is, how does developmental phenotypic plasticity work? It's sort of a fancy term, but really what it means is that organisms with the same genotype can actually produce wildly different phenotypes. We can see some evidence of this in identical human twins, where there's obviously a lot of sameness, but the more you get to know identical twins, the more you can see some interesting traits and behaviors between them. These differences are much more exaggerated in many other animals, especially invertebrates. And I have a few classic examples here. So social insects, those different castes that we see, queens and workers and soldiers, those castes are not determined genetically, but through environmental induction. Similarly, relatively harmless solitary grasshoppers can become crop-destroying locusts, and that is again an environmental induction as opposed to a genetic determinant. And then one of my favorite examples are these water fleas, which can develop a medieval defensive style helmet when they sense predators, but in the absence of those predators, they lack those structures. And so the important point here is that all of these fascinating differences, again, are not genetically determined, and therefore, by definition, must have some epigenetic component. And the organism that I wanted to study, and we do study to try to address this question, is a roundworm, or scientifically known as a nematode. And the particular species of nematode that my lab works with is called Pristionchus pacificus. This nematode is particularly well suited to study plasticity because of this remarkable trait where adults can form one of two possible mouth forms. In some conditions, these worms will develop a narrow, sort of tube-like mouth with a single flap-like dorsal tooth. That's the one in blue on the left. But if we take that same worm and grow it in other conditions, for example, crowded conditions, that worm will develop a wider mouth, a more cuticularized dorsal tooth, and most strikingly, a second additional tooth. And those worms can actually eat other worms and become predators, whereas the other morph are obligate bacteriivores. They can only eat bacteria. Now, I would be remiss if in a talk that features nematodes, I did not talk about Pristionchus' more famous cousin, Cenorhabditis elegans. So C. elegans is the more famous model organism, and just last week, researchers who study C. elegans were awarded the Nobel Prize in Physiology and Medicine. So Gary Ruvkin and Victor Ambros discovered in the 90s what are called microRNAs using C. elegans as a model system. And these are small, non-coding RNAs that control a suite of important developmental processes and are conserved from worms to humans. So now that I've introduced and given fair credit to C. elegans, this is a video of Pristionchus eating C. elegans. And I'll allow the audience to decide for yourself which of these two worms you think is cooler. Okay, so back to the main story. It's 2020. I'm excited to open my lab using Pristionchus to study plasticity. And some of you, probably many of you, will remember in 2020 there was like this whole thing, starting to trickle out stories about a coronavirus that suddenly became a global pandemic. And because of the restrictions on in-person work, instead of really starting my lab in earnest, I ended up doing a lot of hiking. And to be fair, there is, I think, literally no better place on earth to be stuck outside doing hiking than in Utah. So in many ways, we were fortunate. One of the places that I was really interested in exploring was Great Salt Lake. And admittedly, at the time, Great Salt Lake did not have the best reputation. So in many parts of the lake, the sagebrush sort of covers the perimeter of it. And in certain parts of the year, it is completely teeming with giant orb weep spiders. If you go closer to the shore of the lake, the brine fly swarms are so large that it appears as if the earth itself is moving. And of course, there is the noxious sulfur smell that sometimes wafts over from Farmington Bay. However, I would argue if you go at the right time of day, at the right time of year, the Great Salt Lake is actually a strikingly beautiful place. In Antelope Island, where I did a lot of my exploring, There are wild pronghorn antelope and sort of semi-wild herds of bison. There are also incredible populations of birds, both resident and migratory, including the world's largest population of Wilson's phalarope and eared grebes. And I came to learn that Great Salt Lake is also an important cultural icon. It's the namesake of our city. It also supports, as I just mentioned, millions of resident and migratory birds, and is a fascinating extremophile ecosystem that these birds depend on, and we'll talk a little bit about that later. And importantly, in 2020 and 2021, we started to hear about the Great Salt Lake ecosystem being threatened by overconsumption of water. And there were numerous articles that started to trickle out. This one was probably the most prolifically shared. It was in the New York Times, and it was given this dramatic title, "As the Great Salt Lake Dries Up, Utah Faces an Environmental Nuclear Bomb." And what that article is referring to is depicted in the graph below, where on the y-axis, I'm showing the lake elevation, and on the x-axis, years. And this is from utah.gov. And what you can hopefully see is that on the right side of the graph, towards the 2021 era, the lake is reaching record lows in elevation. And as it reaches record lows in elevation, the biota that lives in the lake is becoming threatened. And perhaps even more perniciously, the lake bed playa is becoming exposed because the water is receding. And the threat there is that the lake bed, as a terminal lake, has received thousands of years of minerals from montane runoff, including toxic metals. And when that playa becomes exposed, if wind kicks up the dust off of that lake bed, it can create toxic dust storms, threatening the metropolis area around Great Salt Lake, including Salt Lake City. So it is an area of immense beauty and immense concern, and I wanted to check it out. So on one of my trips to Antelope Island in Great Salt Lake, which is no longer an island because of receding water levels, I stumbled across this sign, which says, "Brine flies and brine shrimp are the only organisms that can tolerate the high salinity of these waters." And the entire reason I told you about my other study with nematodes is to prepare you for what I was thinking when I saw this sign. So here were the things swirling around in my brain when I read this sign. Nematodes are among the most diverse and abundant organisms on Earth. Each of these amazing, beautiful beasts you see here is a nematode. There are at least 25,000 known species, and the estimates of the undiscovered species range up to the millions. They're found on every continent, in nearly every ecosystem. They represent 90% of all animal life on the ocean floor. And perhaps the most striking fact is that on the terrestrial biosphere, 80% of all animals is a nematode. I'll say that in a different way. On land, 4 out of every 5 animals is a nematode. They're also found in extremely cool and extreme places, including an abandoned mine miles below the surface without any access to sunlight, in the McMurdo Dry Valleys of Antarctica, which is described as the coldest and driest place on our planet. And recently, in 2019, a paper published a report of multiple nematodes found in Mono Lake, which is a saline terminal lake, arsenic-rich, and it has some similarities to Great Salt Lake's environment. So these were the things that I knew about nematodes when I read this sign, and so naturally I thought, well, let's see about that. So I went out with my bucket and my shovel, and I started sampling around the lake. And I collected roughly 25 different samples in different areas. I brought them back into the lab, and I tried to extract them with a very common method called a Behrman funnel. It's very simple. You're seeing it right there. And I found 0 worms. Not waiting for the slide to advance, I did not find any worms. And I was humbled by this experience. And I thought, okay, maybe that sign was right. Maybe it is just too extreme of an environment. But I had one extra card up my sleeve, which is I knew that the scientist who studies nematodes in Antarctica is actually a professor at BYU, just down the road. And so I wrote to this guy, Byron Adams, and I said, hey, I've got this idea for a project. You find worms in the coldest and driest place on Earth. Can you show me how you do it? And so he was very gracious. He invited me down to his lab, and he showed me this method, which I won't go into detail here, but it's called density centrifugation. And it's used when you're looking for nematodes in low population density areas. So armed with this new technique and a little bit of chutzpah, I came up with a new sampling strategy that began in the freshwater Weber River and gradually moved into the lake. And the idea here was that if we really can't find worms in the Great Salt Lake, we'll at least know when we stop finding them. And so then we might be able to find the salinity limit for nematodes, which might be useful in and of itself. And so this is just some pictures of us going out on that trip with masks, and this is, I wish I knew what I was saying in the bottom one there, but I like to say that's me telling my lab the chances of us actually finding anything. So these are just pictures of some of our sample sites, and in the lower panel, what I'm showing you is the salinity of our different sites. So you can see in sites 1, 2, and 3, very low salinity, freshwater, and in sites 3, we're starting to get a little brackish. But in sites 4, 5, and 6, which are in Great Salt Lake proper, you have got really high levels of salinity. And for reference, the top orange dashed line, I know you can't read it, is the salinity of the ocean, and the lower dashed line is the salinity of Mono Lake. So we are in a completely new, different salinity regime here in Great Salt Lake. And the punchline, which you probably predicted, otherwise I wouldn't be here talking to you, is that we found living worms. And this was really one of the most exciting sort of discoveries that I've been a part of. I remember fondly that we were out in the lake the entire day. We were hot, we were tired, but we wanted to process our samples right away. So we came back and we extracted the worms, and it was around 9 p.m., and we're all huddled over our microscopes. And eventually one of my grad students kind of yelled out, "I found one!" And we all ran over and looked at her microscope, and indeed we saw a worm. And another 5 minutes passed, and somebody else shouted out, "I found another one." And over the course of the next hour, we found probably about 20 worms. And you know, this, in our little corner of the world, this was just the joy of scientific discovery, where we went home that night knowing that about 4 people on this whole planet know that there are worms in this lake. And it's a really the fun part of being a scientist. Okay, but we wanted to know more about these worms. And so this is a very simple, but I think very powerful experiment where we wanted to see how adapted the worms were to the Great Salt Lake environment. So we simply put Great Salt Lake nematodes and laboratory nematodes, C. elegans and Pristioncus, in water from the Great Salt Lake. And not surprisingly, Great Salt Lake nematodes did fine, but worms, the laboratory worms, C. elegans and Pristioncus, died almost instantly, indicating how toxic that salinity is to their physiology. And I think fascinatingly, the reverse was also true. When we put Great Salt Lake worms on standard growth plates, which of course C. elegans and Pristioncus do completely fine in, they died, Great Salt Lake nematodes died instantly as well. And so this, again, simple experiment demonstrates that these worms are very adapted to this environment, and sort of implies that they've been out there long enough to adapt to this environment. Something else that we noticed straight away was that in the hypersaline sites, there were really very few worms. So we rarely found more than 5 worms in 100 grams of sediment, in contrast to the freshwater sites where we'd often find 200 worms per 100 grams. And however, there were occasional samples where we would find dozens of worms in hypersaline sites. And so we began to wonder if there was some sort of microhabitat that these worms might like to live in. And we wondered if we could detect a chemical signature of that microhabitat or environmental niche. And so we collaborated with Diego Fernandez in the geology department to do inductively coupled plasma mass spectrometry on our samples and try to find a chemical signature that would correlate with nematode abundance. So what I'm showing you on the left here is the chemical profile of all of our different sites. And what we saw was sort of what we expected, in the sense that all of the warm colors are the hypersaline sites, and they all cluster together. And all of the cool colors are the freshwater sites, and they all cluster together. But sort of sadly, disappointingly, we did not observe any real strong positive correlation between any element or compound that we measured and nematode abundance. So we kind of went back to the drawing board, a little dismayed. And the answer to this riddle, actually, in hindsight, was really looking at us right in the face the entire time. So this is a picture of me on one of our sampling trips, and we literally had to navigate our boats around these rocks on Antelope Island, or on the edge of Antelope Island. And it turns out that these are not just rocks. These are fascinating living rocks built by bacteria. And over time, bacteria and algae can sort of precipitate calcium carbonate in situ and form a dense rock layer, and then a new layer of bacteria and algae form on top of it. And that's how you get these increasing size mounds. And so they're actually living rocks called microbialites. And now this is the point in the story where I have to introduce the hero of this next breakthrough, which is Julie Jung, who was my postdoc. This is a picture of her out at a sort of endless field of microbialites. And one day I was in my office, and she came in and said, "Michael, I think the worms might be inside the microbialites." And I thought, oh, that's a very interesting idea. And I kind of sat back in my chair and stroked my imaginary beard. And she went back into the lab. And I'm imagining designing an experiment to test this hypothesis, which would probably take about 2 months to do. And as I'm thinking that, I hear this tremendously loud banging coming from the lab. And so I ran into the lab thinking maybe a centrifuge was going to go through a wall, but there was Julie with a hammer pulverizing one of these microbialites. She then took that pulverized microbialite and put it through our density centrifugation method, and she found hundreds of nematodes. And this was really, again, a dramatic breakthrough for us. We did end up going back out and doing that longer, more carefully controlled experiment, and it verified or validated Julie's first result. So if we look on the y-axis at nematode abundance, and it's on a log scale, we see many more nematodes from microbialite samples, often in the tens, sometimes in the hundreds, and occasionally even thousands of worms in 100 grams of sediment. And in neighboring sediment just next door, again, we rarely find more than 5 worms. And so what we now think, our current hypothesis, is that the microbialite provides an environmental niche rich in bacteria, which serve as food for the worms, and it may even protect them from UV or aridity that they would otherwise face in the high desert environment of Great Salt Lake. And so we think they might be a refugia of sorts for these worms. And the panel on the right, I'm showing all the places in the lake where microbialites have been mapped. So I hope you can appreciate that there are probably in the millions of worms surrounding this lake. And I want to plug microbialites one last time because of how fascinating they are. This is a paper, among many others, that have found fossilized microbialites dating back more than 3 billion years. And so we actually know when life began on Earth because of fossilized microbialites. They actually were the dominant macroscopic evidence of life on Earth for billions of years before anything else arose. In the last 500 million years or so, they started to decline, and only a few places in the world still contain them today. And our Great Salt Lake is one of those places. So right in our backyard, we have this amazing ancient ecology, and we've got this rare opportunity to study it. And we now know that nematodes are part of this story. Okay, I'm going to take a quick break because I know attention spans are short. So I'll just drink a bit of water. You can stretch your legs. I'll take a couple questions if anyone has one before we get into the next phase of the talk, which is focused on trying to identify who these worms are. Yeah.
Is it possible for the nematodes to have potential predators in the Great Salt Lake?
Yeah, so the question is it possible that some nematodes have predators in Great Salt Lake? And the answer is it is definitely possible. We don't know yet, but we have looked to see if brine fly larvae or brine shrimp might be eating them. So far, there's no direct evidence, but we haven't ruled it out.
Was there ever a period of time where like microbialites were much more prevalent, or have they always been like more rare?
Yeah, so that's what I was trying to say near the end, that for most of Earth's history, that life existed, so about 3 billion years, the major form or evidence of life were microbialites. And it's only recently that they've really declined.
Hi. Do you think the nematodes have any role in building the microbialites, or is that just the bacteria?
Yes, I do think it's possible that they have a role in building microbialites. And this is one of the more exciting avenues of research that we're trying to follow up on, because these microbialites are very porous in nature, and it's not really understood how or why they're porous, but nematodes are known to tunnel through sediment and carry bacteria with them. And in the absence of that tunneling, these microbialites might look completely different. So we're trying to think of ways to test this hypothesis, but it's certainly one we're interested in. Yes.
Oh, stand up. Can I go? Okay. Oh. Okay.
Well, so the worms that we used for that experiment that were that died in the Great Salt Lake water, those were just standard laboratory worms, so C. elegans and Pristioncus. So they've evolved many millions of years away from these guys. Yeah.
Oh, perfect. And then what about those nematodes from the Great Salt Lake in normal media without the high salinity level? Did the two different populations from high salinity levels and low salinity levels behave any differently?
Behavior-wise, we haven't measured too much behavior other than dead or alive. So to be determined. Yeah.
Thank you.
Yeah. Okay, I'm gonna hold off on the remaining questions and get to the rest of the lecture just so that we stay on track. But please keep your question and we'll get to it. Okay, everybody ready? So, one of the questions that really drives our current research, and I'm excited to peel the curtain back and let you in on what we're really doing now, is to try to identify who these worms are. What species are they? Where did they come from? Who are their closest relatives? And if we can answer those questions, maybe we can start to get some insight into where they came from, which is, to me, a fascinating question. especially given the incredible history of Great Salt Lake, many of which you know that about 30,000 years ago, this entire area was inundated with a freshwater lake. Well, as I just showed you, these worms would not survive in a freshwater lake. But prior to that freshwater lake, there was about 800,000 years of a similar environmental condition to there is now. So did they evolve then, somehow survive the freshwater lake, and then readapt to salinity? Or did they come from somewhere else? Well, there's nowhere else within thousands of miles that has a similar environment. So these are the questions that are really driving a lot of our current research. Okay, so how do we tell who these worms are? We can do this using a little bit of molecular biology, where we pluck worms and put them into a buffer that, sadly for them, essentially melts them. And then we can do PCR to amplify certain regions of their genome and then sequence those regions of their genome. And okay. So this graph is a little bit complicated, but I'm going to try to walk us through it. Each brick represents an individual nematode. And what I'm showing you is the color corresponds to a nematode family, shown on the right. And on the Y is the percent family found in each of our 6 different sites. And so what I think is very sort of obvious is that in the freshwater sites 1, 2, and 3, there's really a diversity of nematode species out there, more than a dozen different families, which is really incredible if you think about it, because it's like a handful of dirt harboring dozens of different families. But that diversity falls off a cliff once we get into the hypersaline sites. There's essentially only one family that these nematodes belong to. And just to give you a little bit more insight, so family on the right is a taxonomic level below order but above genus. So what family do these worms belong to? The family is called Monohistoridae, and this turns out to be an ancient and widely dispersed nematode family that's found in almost every aqueous environment. And particularly interesting to me, they are the dominant animals found on the bottom of the ocean and near deep sea hydrothermal vents. So they seem to have this remarkable ability to adapt to extreme environments, but we really don't know much more about this family other than their unique ability to colonize various aqueous environments, now including also Great Salt Lake. If we take that sequencing data and put it into a phylogenetic tree, all I want to convey to you is that I colored the branches of the tree red that came from Great Salt Lake, and all the branches that were in blue came from the freshwater sites in the Weber River. And so what I think you can appreciate is that the Great Salt Lake nematodes really form their own distinct clade. And this implies, although we don't have enough resolution yet, but it implies that this is a newly discovered species. And that leads towards our next question: are these indeed a new species? And so to answer that question requires a morphological description of these worms, and that's one of the things that we're actively working on right now. And so just peeling back the curtain a little bit to show you a little bit of what this looks like, we're taking a lot of images of nematodes using the microscope, and we're starting to identify anatomical features on them so that we can compare the micrometer positions of those features to other nematodes within the family. And at some point, we'll be able to say those anatomical features and morphologies belong to this species, or if they don't belong to any known species, that's when you can declare this is a new species. These are some recent, I think, beautiful images. People have told me they're scary, but this is a scanning electron microscope image. So light microscopy has its limitations based on the wavelength of light. But if you want to get better resolution, you can use electrons, which have a much shorter wavelength, and therefore more resolution of the specimen you're looking at. And so we can see the incredible detail of these small microscopic organisms. And again, we're using these images to compare the morphology of these worms to other monohystridae nematodes. And this is really a fun puzzle, in fact. And so I'll just give you a brief, very quick insight into how this works. So we start looking at a bunch of taxonomic keys. And when we look at females, we came up with this, checking off these various taxonomic keys, and it looks like the species belongs to the genus Eumonohystra. Then we started looking at males, and that changed the picture completely. So we can see double spicules in the bottom here, and these fin-like, what are called bursa, that are on this is like the tail of the worm, that extend around the cloaca. And those features sort of unequivocally put this worm into the genus Diploomyloides. And so we've now got these two different sort of interpretations of this data. And I recently spoke with Gustavo Fonseca. He's a Brazilian scientist who came up with the taxonomic keys. And I showed him the female data, and he said, great, looks like Eumonohystra. And then I showed him the male data, and he said in this beautifully accented Brazilian English, no problem, Michael, I've got an easy solution for you, just delete the male photos. But we do think the male copulatory organs are much more easy to identify and hard to evolve, and so we do think these are probably Diploomyloides, but we don't know yet, and we're actively trying to figure out, again, if it's a new species within that genus. Okay, so to summarize, we have found, credit mostly to Julie Jung, that nematodes do live in the Great Salt Lake, Utah, and it is truly one of the most salient environments that any animal is capable of living in. They're highly enriched in microbialites, and this is a new environmental niche that worms have been found in, and we think they serve as a refugia for these nematodes. And then finally, this is actively ongoing, but we sort of hypothesize that these are new species within the genus Diploomyloides. And these are publications that we've put out that describe this work in that more formal, this is exactly what we plan to find, and this is how it unfolded, if you want to check those out. And so finally, I'd like to acknowledge, again, Julie, for leading this work. I'd also like to highlight Maxime Razul-Agha. He's an undergrad who's on this paper, and he did some really fascinating work with the bacteria from microbialites and the worms. And then finally, Morgan Marcu is a new grad student in my lab who's taking on this project and doing a lot of the taxonomy as well. I'd like to thank Diego Fernandez for helping us with the mass spectrometry, Byron Adams for showing me how to find worms in low population density areas. Abigail is his grad student who's done some help clarifying some of the taxonomy for us. And then finally, Tom Power's group at Nebraska for doing the scanning electron microscopy. And of course, thank the funding resources, and would be happy to take any more questions should you have them. Thank you.
This for me so I can read it. Okay.
Hi. Have there been nematodes found in microbialites in other parts of the world?
No. Well, not that's been published, but recently I got an email from a grad student in China who said they saw our paper and started looking at microbialites in an inland lake there that had microbialites, and he sent me back a photo of one. So the story is probably more common than just Great Salt Lake. Great question.
Hi. Could the lab nematodes that weren't the ones that you found in the Salt Lake, could those live in the microbialites that you found in the Salt Lake?
So imagining a scenario where we sort of seed microbialites with those worms. in a backwards way, we have done that experiment, and the answer is no. The laboratory worms die instantly as well. And it seems like it's because those microbialite pieces or chunks are also super saline as well. I can give you a more detailed answer that leads to another sort of research direction, but I'll leave it at that for now.
Hi. Hi. So I was curious, with the extreme salinity of the Great Salt Lake and all of those other factors, is there also a possibility for tardigrade life? Because I know they can live in very extreme environments as well. So I was just curious about that.
Yeah, fun question. We have seen tardigrades in the freshwater sites, and they're super cute, and they're wriggling around. These water bears, if you don't know what they are, look them up. They're amazing. But I will say proudly that they do not survive the salinity of Great Salt Lake. Only our nematodes do.
Has there been any efforts to like dating the microbialites, and if so, has there been like barriers to that with like erosion or just the growth of the new layers?
Yeah, so it's a great question. There have been some efforts to date the microbialites in Great Salt Lake, and the best estimates are that the big ones could be 10,000 years old. But it's really hard, and the reason that it's hard is that, as I mentioned in answer to an earlier question, they're porous, and so new fresh water or minerals can get into the deeper layers. And so it turns out it's not trivial to just take a sediment core and say the bottom is older than the top. We are thinking about ways to try to get at that with some collaborators, but probably the big ones are thousands of years old.
Since there have been nematodes found in the Great Salt Lake, have there been any found in other salt lakes around the world? Or are there people trying to find them?
Yeah, so as far as I know, this is the most saline lake that they've been found in. But again, I mentioned Mono Lake, which is far less saline. You can find worms. And I know in Patagonia in South America, people have also found lakes, worms, nematodes in saline lakes. Interestingly, they belong to the same genus as our worms. And so one of the, I think, exciting things that we're doing next is we hope to travel to Patagonia, sample some of those worms, and see how closely related they are.
Hi. Is there any potential to learn about the ancient ocean that used to be in Utah through nematode studies?
Excellent question. So my brain hasn't gotten quite to that sort of research avenue yet, but I will just highlight what the basis of the question was that millions of years ago, this was an inland ocean. And so maybe that's when the nematodes finally got here and then continued to adapt as the basin became what it is. But yeah, I don't really know.
You said that the microbialites kind of acted as a refuge for the nematodes, but originally you found them in the actual lake itself. So would you say that in different parts of their life cycle they would kind of move between, or are they almost like separated communities? Yeah,
so I think that when we find them in lake bed sediment that's not microbialite, that they're sort of transporting or migrating around the lake. But their main habitat, if you will, is in these microbialites. But one of the ways that we can try to get at that question, which we haven't done yet but are eager to do, is sample really different places across the lake and look at the genetics. How similar are nematodes from one section of the lake to the other? If they're vastly different, it would suggest there's not that much cross-pollination between them, or not a lot of migration. But if they're very similar, it would suggest that they are traversing fairly large distances, to be determined. Great question.
General nematode question. Do we see them exclusively in natural habitats, or can they be found in like macroscopic organisms like whales and stuff as like a parasitic or symbiotic relationship?
Yeah, so we find, so nematodes can broadly be categorized into two different groups: free living, like the ones I talked about today, but also parasitic nematodes. And there are some that seem to live an intermediate life where they transport to other places on insects, but aren't necessarily a pathogen of them. But you brought up the whales, so I'll highlight, in case some of you might have seen the worm wrangler video, that the largest nematode known to exist is about 30 feet long, and it lives in the placenta of a sperm whale.
With the changing conditions around the Great Salt Lake in the last couple of years, do we know if there's implications for the nematodes?
Yeah, so another great question, and I do think, sadly, the answer is yes. So when we sample from desiccated, exposed microbialite above the water table, we find far fewer nematodes, and they look like they're one foot in the grave, the other on a banana peel. When we sample from really healthy-looking microbialites under the water table, that's when we see a lot more worms, and they look a lot healthier.
Okay, so like the Great Salt Lake varies in salinity, so I was wondering if like you're looking at like how they vary in different salinity areas and whether or not they can live in different salinity than the ones they're found in.
Right. So I think between probably 9 and 20%, they're fine, and that is really a huge range. So quite a feat. Below that, they don't seem to tolerate. So 5%, I think, is probably the limit at which these worms can survive in. The north arm of the lake is really the supersaturated, where you actually see salt crystallize, and that's approaching 35% salinity. And we have not found worms in the north arm, and probably that's too extreme. Great question.
Do the nematodes in the Great Salt Lake have the same ability where in the populous areas they become omnivorous, whereas in the less populous they remain herbivorean, or is that completely separate, a different type of nematode?
Yeah, fun question. Most likely no. That predatory to omnivorous or predatory to bacterivorous plasticity seems to be a evolutionary novelty in the family Diplogastridae, of which Priscionchus pacificus is part of. But it seems to be a novel innovation in that clade of worms.
Oh. Well, what was up with the male parts? There was like 2 lines. What did that mean?
Yeah, so that's a double spicule, and to put it in layman terms, they have 2 penises.
Oh.
And that's a very unique feature of this genus, and so it's pretty clear that is the genus they belong to. I assume I answered your question.
Yeah.
I had the same.
If nematodes were ever to become endangered or extinct, how would that affect us and where we live?
Another fun question. So I don't want to run that experiment because they are so ubiquitous and so important in nutrient cycling and in decomposition. It's been postulated if you ran that experiment, within a couple days, you would the stench of non-decomposing life would become horrific. And the entire ecosystem of our planet might change because, again, they play such an important role in nutrient cycling and decomposition.
Yeah, so how would you determine a holotype specimen with the males and the females being so sexually dimorphic?
One of each. So we would have one holotype of a female and one holotype, and several paratypes of each. And that's exactly one of the things that we're working on. And those will be vouchered hopefully soon at the Natural History Museum.
Sorry, just over here. There you go. What would these nematodes eat?
in the Great Salt Lake?
Yeah, so we're very confident that they eat bacteria. We can actually see in some images bacteria in the sort of esophageal lumen of them. They might also eat fungi. That's another sort of hot area that we're looking into, but we know they can eat bacteria.
Sorry, that was really loud. You mentioned earlier that a lot of your work also had to do with like epigenetics. Does that relate to anything that we talked about right now?
Not directly, but because that is sort of where the other half of my brain in the lab lives, when we are able to hopefully someday culture these worms in the lab, which we're making progress on, I am interested in doing some experiments to see if you start them out as larvae or juveniles on a high saline environment, does that mean they switch their physiology to be more able to do that as adults compared to reversing the situation? But that would sort of be a fun way to connect those two stories, parts of the lab. But right now, there's no direct connection.
So when you were talking about the way that the environment of like the nematodes, how before it was like a freshwater environment, and then now they're living in a very salty environment. Do you think that occurred because the environment gradually became more salt, have like greater salinity, or was it because I know that in like the oceans they have like systems where they have like deep pools at the bottom of the ocean that are very salty and are colder than the entire ocean? Maybe is that like a phenomenon that can happen in freshwater environments?
Oh, super interesting question. I unfortunately do not know the answer to that, but I think it's a fascinating idea. So maybe there was a reservoir, a deep pool of salinity that remained even when Lake Bonneville was here, and they hid away there and reemerged when Lake Bonneville disappeared. Fascinating idea. No idea if that's possible.
All right, I think that's it for questions. Maybe we can thank Professor Werner.